Hey guys, welcome to Chapter 8, Topic 3. In this topic, we're going to talk about DNA recombination and what that means for microbes. This topic is important because this is how bacteria share genetic information, and this is how they can pass along traits such as drug resistance, which is a big problem in the hospitals. We also use this information for biotechnology applications, which we'll talk about in a few topics from now. So let's start off with what is DNA recombination. Recombination is when the DNA sequence of an organism is changed. There's natural recombination events in which one bacterium will donate DNA to another bacterium, and that'll get incorporated into their original genome, so it'll change the way that they behave either through like a drug resistance or maybe a phenotypic change, things like that. Or even metabolic processes. One bacteria may be able to metabolize a sugar that they then pass to another bacteria. There's also artificial recombination, and this is what we do in the laboratory, and we'll talk about this in the genetic engineering topic. But we are able to make bacteria take up new pieces of DNA to start production of something that we desire or want them to do. So the big thing in genetic recombination bacteria is called horizontal gene transfer. So what is horizontal gene transfer? This means that we are transferring genetic information from one adult cell to another adult cell. This is in comparison to vertical gene transfer, which is what we see most often because we don't transfer genetic information between each other. But in horizontal gene information, we pass that from parent to offspring to offspring to offspring. And that's called a vertical gene transfer. So remember, horizontal gene transfer is when adult cells pass genetic information to each other. Now there's two ways that horizontal gene transfer will occur, either through a plasmid, which remember is a small circular piece of DNA, or fragments of chromosomes. So either one of those can be used to pass that information. Now, one important aspect about plasmids is that they contain their own origin of replication. This means that it doesn't have to be fully incorporated into the host cell's genome in order to be read. So plasmids have a little bit more of an advantage in that aspect. Now, there are three types of horizontal gene transfer we're going to talk about in this topic. We got conjugation, transformation, and transduction. And so let's take a look at each of these now. So first is conjugation. This is sometimes called bacteria sex because this is the only form of horizontal gene transfer where bacteria will actually touch each other, and they transfer genetic information via their sex pili or through a specialized channel depending on the cell type. And your book goes into a lot of information about how the F factor works and all this other stuff. I don't really want you to focus too much on that. I mean, you can if it's interesting. But the big important aspect of this is that it requires physical contact, it creates a bridge, and then the plasmid is then shared between bacteria. Okay, so that you can see an example of how this is happening here with the F plus and the F minus receptors. So look at this and let me know if you have any questions. But as I said, the big thing about this is that they touch and they share a plasmid. Then we have transformation. This is actually an activity you guys are going to do in the lab later this year. The transformation happens naturally as well. This is when a cell will spit out DNA fragments from a donor cell. So they just dump out fragments. There's also a recipient cell that's present, and this recipient cell has to be competent, which means it has to be able to take up these DNA fragments. Not all cells are capable of doing this at all times. So that's one aspect of it. Just because we release these DNA fragments into a solution doesn't mean that all recipient cells will take it up. So it's not 100% transformation rate. So once the cell brings the DNA into the cell, it'll then use that to incorporate information into its own genome and use that to become a genetically transformed cell. So you can see some examples here. So remember, conjugation, they actually physically touch. Transformation, they don't. Conjugation, we use a plasmid. Transformation, we use DNA fragments. So just try to keep them straight. We have one more example of horizontal gene transfer, and this is through transduction, and this is biviral. So we got conjugation where they touch, transformation where they don't, transduction where we use viruses. Now the key aspect about this, we've already talked about viruses earlier this semester. What happens in this virus situation is that the DNA of the host cell gets mixed up with the viral DNA. And so what happens is when these new DNA or these new viral particles are made, there's defective virons made as well. And they're carrying the DNA of the host cell. So they have a little bit of the host cell with them and they don't have the viral DNA, which means that they can't really infect DNA or infect cells the way they normally do. They go and they do travel to new cells and they do inject this DNA, which incorporates it into the cell, but it doesn't infect it and cause delyse, which is how this genetic information is then carried forward. So let's review these again. Conjugation, they touch, they share plasmid across a pili or a channel. Then we have transformation, where DNA fragments are taken up by the donor cell. And then we have transduction, where information is passed via defective bacteriophages. So make sure you understand those three components. As I said, I'm not too worried if you know the nitty-gritty details about how each of these are, but I want you to know the overarching ideas that are associated with each of these and be able to tell them apart. Now we have one more thing, and this is not horizontal gene transfer. It can become part of horizontal gene transfer, but it's not on its own. And these are transposons. Transposons are jumping genes, and we actually have these ourselves. These are genes within the genome that can move around. And what'll happen is it can jump inside a gene and cause that gene to turn off. It can jump out of a gene, causing a gene to turn on. It can cause a change in the way the gene is expressed. And so all these things can have an impact on how the bacteria works. And these, as I said, these transposons exist in almost all species that we know about. And there's still a lot of argument as to how these jumping genes have come about. But it's important to be aware that there is some recombination that can occur within the cell itself. Now, if this gets passed through any of the three methods we just talked about, then it can become a vertical gene transfer. So those are the main concepts in DNA recombination within microbes. Please review your objectives and let me know if you have any questions.